Hello, I'm Pinakin Davey, Professor of Optometry at Western University of Health Sciences, and it is indeed my pleasure to welcome you to this video series. How do I correlate structure and function in glaucoma? Where glaucoma is concerned, the structure and function should correlate to some extent. The ganglion cell dies, and so you have a visual field defect. But that's not always the case, as we know. Let us look at some examples. So here's a visual field plot, right eye and left eye of a patient, and without looking too much at it, we know that there is a stark difference between right eye and left eye of this patient. So you can see less defect, more defect. Now you look at retinal nerve fiber layer using OCT, looking at the nerve fiber layer plots, we can hardly tell the differences between right eye and left eye. Let us look at the visual fields again. Stark difference, almost the same. Well, what happened? Well, we do know from various studies, and here I am showing a graph from Felipe Medeiros' paper. You have estimated retinal ganglion cells and mean deviation along y-axis. In early glaucoma, what we find is that the ganglion cells are dying at a steady rate, and the visual field is almost stable for a little while. So there is a discordance early on in early glaucoma, but for majority of the disease, they tend to run parallel, that you're seeing that the visual field damage sort of mirrors or parallels itself to structural damage that is visible. And towards the tail end, what we find is that the ganglion cell loss is barely visible, or it becomes flat and it's not visible at all, whereas there is a stark, fast decrease in visual field. So except for the tail end and very extreme early glaucoma, one should expect a reasonable correlation in structure and function. Ted Garvey-Heath did a fantastic job of explaining which parts of either rim tissue or nerve fiber layer correlates appropriately with the visual fields. So you look at this purple color, and this is the inferotemporal region of the optic nerve or retinal nerve fiber layer that should coincide nicely with this superior visual field. Or you look at this orange-like color, supratemporal region, that should coincide nicely with inferior part of visual fields. You get the idea. Octopus visual field has something called a polar graph. Polar graph is very helpful in trying to give us an understanding of structural function relationship, and as well trying to locate region or explain to us which regions in the optic nerve or nerve fiber layer should we expect a problem. So if you see a visual field deficit, the larger the deficit, one would expect a larger polar graph line. So you can see here, this is showing a defect, and you see a corresponding polar graph defect. So here, let's start with this. A paracentral defect, a nasal step, should coincide appropriately with a region in the optic nerve, and that's your polar graph. And here you're looking at HRT printout that says that you should expect a deficit or a damage in the nerve right about here. So you have a normal visual field progressing in glaucoma. One would expect the polar graph to be getting longer and longer as this damage is observed. Thank you very much.